In this video, we will discuss about role of tumor-derived lymph node innervating neuron in suppression of anti-tumor immunity. So, in order to check this hypothesis or find out the role of this innervating neuron, what they did is they took this donor mouse that is infected with the tumor and take out this tumor-derived lymph node. From that tumor-derived lymph node, they sorted out the immune cells using these CD45 plus markers, and then they co-culture and they intratumorally inject these immune cells or the conditioned media or the inactivated conditioned media into the lympha lymphadenectomized recipient. This means this mice does not have lymph node, and then analyze the tumor volume. So when they analyze the tumor volume in the different conditions, what they are able to see compared to the control, which does not have the immune cells, just the saline intratumoral injections, the immune cells cause reductions in the tumor volume, indicating that these immune cells are necessary for tumor suppressions, or this helps in the suppression of anti-tumor immunity. And these things, the tumor cell suppressions will be seems to be enhanced when they use the chemogenetic reagent that results in further tumor suppressions that can be seen in the panel B. And similar thing they have tested with conditioned media and heat activated inactivated conditioned media. In the conditioned media they see the similar results, that is with the immune cells only they are able to see reductions in the conditioned media from the immune cells. They are able to see, without any treatment, they are able to see tumor reductions significantly compared to the control. The tumor reductions, they do not see the tumor reductions are significant. Yeah, that we can see that. However, when it is clubbed with the chemogenetic reagent, the tumor suppression is statistically significant compared to both control and the immune cells conditioned media only. And when they use the heat inactivated conditioned media, they do not see the difference in even in the presence of chemogenetic reagent compared to the case of the conditioned media, indicating that proteins that are secreted from the immune cells or from the tumor-derived lymph node containing immune cells helps in this tumor suppressions, and that can be inhibited by inactivating those proteins using heat. This is what they did in inactivated conditioned media. Afterwards, they checked what are the genes that are upregulated and downregulated using gene ontology analysis of the stereo-seq of this tumor-derived lymph node and also the single-cell RNA-seq data. Combined together, they are able to find out that the top upregulated pathways include T cells activation, alpha beta T cells activations, adaptive immune cells response, gamma delta T cells activations, and the paths that are downregulated is related to the response to ER stress, B cell activations, aerobic respirations. These things are downregulated, indicating that the gene pathways that are related to the immune activations are upregulated, and the pathways that are related to immune suppressions are downregulated. And afterwards, they also checked. They also validated it using using the tumor microassays like plates, tumor microassays like system, where they checked it for the different immune cells, that is CD8 plus T cells, which has a direct cytotoxic effect against the tumors, and this uh, dendritic cells, and also the co-localization of dendritic cells and the CD8 plus T cells. So in the compared to the control case, in all the three chemogenetic, all the two chemogenetic agent and the one where CGRP has been knocked down, CGRP protein are been suppressed using Calca knockdown, they are able to see enhanced in the CD8 plus T cells compared to the control, and also enhanced in the dendritic cells, and the localizations can also be seen that dendritic cells and the CD8 plus T cells are found to be localized in most cases. This in all together indicates that there is a role of this innervating neuron in the tumor-derived lymph node, as it will uh, as in the suppression of anti-tumor immunity, because this innervating neuron will results in this formation of results in the formation of these immune cells, and these immune cells will then helps in the tumor suppressions. However, in the case of this innervating neuron, uh, what will happen? For example, in this CGRP will prevent this will prevent the suppression of anti-tumor immunity, and that we can see that in the absence of CGRP, there is an increase in the hydroxy effect. And similarly, with the in the absence of chemogenetic reagent, this innervating neuron can result in this suppression of anti-tumor immunity, as the immune cells will go down in the absence of chemogenetic reagent also. So this in all together indicates the role of innervating neuron in the suppression of anti-tumor immunity. So that's all for this video. If you need to understand more about these experiments in detail, you can go to the reference that has been provided in the description. Thank you for your time.